We will have then Dr. Dinesh Yadav with the concerns in regards to patient safety in the day-to-day clinical practice in acute care setting. And we will have Dr. Malak Al-Mashali from Saudi Arabia with the laboratory perspective, patient safety in blood gas testing. At the end of the presentation, we will have a roundtable discussion among the experts, and we will have our Werfen medical directors as well, with Dr. Klaus Görlinger, our medical director for patient blood management, and Dr. Mark Rubinstein, medical director for whole blood hemostasis. And so few technical notes before starting the webinar. So please feel free to write your comments or questions in the chat box, and we will review the questions and comments at the end of the presentations within the roundtable discussion. After the attendance, a certificate will be available to be downloaded through our Werfen EMEA Learning Center, and the proper link will be sent after the webinar ends. And for the registered participant from Saudi Arabia, since we are cooperating with the Saudi Society of Clinical Chemistry, the Saudi Commission for Health Specialties, and Point of Care Testing Saudi Association, there will be CME credits. So let me start this session, this webinar, talking about patient safety in vitro diagnostics in acute care setting at the point of care. So in particular, let me start with the WHO, so the World Health Organization Global Patient Safety Action Plan for 2021-2030. So this global action plan has been built by the World Health Organization together with the Patient Safety Organization, and it has been published 1 month ago, so in August 2021. And it is a strategic and coordinated approach to patient safety with the objective of eliminating the avoidable harms in healthcare. Within this document, they address the common causes of potential harm and the ways and approaches on how to prevent that harm. So this action plan is built with some framework for action, and in particular in 7 strategic objectives. The first strategic objective is make zero avoidable harm as a state of mind, while the second strategic objective is to build high reliability health system to protect patients. The third one is to assure the safety of every clinical process, while the fourth one is to engage and empower patients and families, involving them in the process of care. Then the fifth one is to inspire, educate, and at the end to train the healthcare workers. The sixth one, ensuring a constant flow of information and knowledge as for the mitigation of the risk and reduction of that risk. And the seventh and last one is to develop and sustain a multi-sectorial, multinational synergy and partnership as a way to improve patient safety and the quality of care. So those 7 strategic objectives have been then declined into actions, and in particular into 35 actions. And the development and implementation of the action plan has patients and families involved as partners, involved in the process of care. But then, of course, taking in consideration a collaborative working between healthcare professionals, always respecting and taking in consideration the peculiarities of the and the nature of the care setting. Always analyzing and sharing data in order to generate further learning. This can be done not only sharing data and analyzing those data, but also with the evidence, and in particular translating that evidence into actionable and measurable improvement, using both that scientific expertise and, again, patient experience. And in the end, all of this is done to build a safety culture, as a safety culture in the design and delivery of healthcare. As you can see here in this infographic that is inside that document, the 7 strategic objectives are at the base of this process and of this overall objective and common goal, while patients and families are involved as partners and are acting as pillars of this overall common goal. And we have the safety culture, again, as a strong pillar of this common objective. But who are the actors? So who is actively involved in this action plan? Of course, there are the healthcare professionals and the healthcare services, and the stakeholders, so the companies and the scientific societies working with them. So that's the reason why we are here talking about patient safety in vitro diagnostics in particular, in acute care setting and at the point of care. Because as Werfen, we are constantly committed and developing the highest quality test results and the most innovative solutions, because we are committed into helping the healthcare organizations in improving patient care. And to do this, we are working together with the healthcare professionals. And in particular, to achieve such an objective of improving patient care, we are working across the hospital continuum. So from the clinical laboratory to the hospital acute care setting. Always taking in consideration the synergies between these departments and working collaboratively. So talking about patient safety and in particular reducing those avoidable harms, let's talk about which are the problems in particular. And we can consider, for example, the transfusion practice. So we can take in consideration red blood cell transfusion because around 60% of allogeneic blood transfusions standing on scientific data published in literature are inappropriate and should be avoided. While if we talk about plasma utilization, we know that up to 58% of plasma transfusions are avoidable, and 25% of those are unnecessary. That means that they have no beneficial effect on hemostasis. While on the contrary, we know all the risks related to transfusions. If we talk about the variability in patients' response to therapy, we can talk about, for example, the antiplatelet agents, because again, from data from scientific literature, we know that at least 1 in 3 patients that are under antiplatelet therapy is not receiving the intended physiological effect. And such an inadequate response may lead either to thrombosis or to an increased risk of bleeding because of the hyper-response to antiplatelet therapy. If you talk about the anticoagulation strategies during the invasive procedures, such as the cardiopulmonary bypass in the cardiovascular operating room, we know that there can be some kind of problem in heparin dose optimization because it depends on several aspects and hypothermia among them. So we know and we have to talk about patient safety at the point of care as improved patient care. And to reduce the avoidable harms, it means reducing the complications at the end. And we can do this reducing, for example, the inappropriate blood transfusions and the clinical complications that are going together. For example, with the use of rotational thromboelastometry guided patient blood management. Or we can reduce the red blood cell transfusion using the hemoglobin monitoring with co-oximetry. Or we can use the anticoagulant dosage optimization through the ACT results. Or again, we can identify those patients who are exposed to an increased risk of bleeding or an increased thrombotic risk because of the antiplatelet therapy. But it is not enough because we know that the other side of the coin is also to reduce the risk of medical errors. And so there can be this real-time automatic quality assurance before, during, and after sample measurement. Then we are moving more and more towards a virtual world, so we know that connectivity is a key. And as stated by one of the strategic objectives, it is important to ensure a constant flow of information and data. And this can be done with a solution that allows the management of information analyzers and operators, with a single unique database to access patient samples and historical results. Again, always taking in consideration the synergistic approach at the point of care. But another strategic objective was regarding training and education. And in particular, talking about the continuing medical education, we can talk about, for example, an online e-learning portal that can be useful for a manager to assign and monitor learning paths, or by the staff directly as a training service for the on-site education. Then I was mentioning before the Werfen MEI Learning Center, the website in which you will find the certificate of attendance. And inside the Werfen MEI Learning Center, you will find the most recent scientific webinars, not only for acute care diagnostics, but also for autoimmunity and hemostasis. And there is also a literature section with the most updated scientific papers. So with this, I thank you all for the attention, and I would leave the stage to Dr. Guttorm Bratebo from Norway, talking about the 10 years of the Helsinki Declaration on Patient Safety in Anesthesiology. Please. Doctor, maybe you're on mute. Can activate. Yes, thank you.
There we are.
Yes.
Thank you, Valentina, and thank you for your introduction. And also thank you to the organizers for inviting me to speak on patient safety, and specifically the Helsinki Declaration on Patient Safety in Anesthesiology. I will share some slides with you now. Can you see it?
Yes, not in presentation. Okay, perfect.
Are we there?
Yes.
Well, thank you. I'm working at Haukeland University Hospital in Bergen. And Bergen is the second largest city in Norway and located on the western coast of Norway. And my declaration of interests are that I've been member of the board of European Board of Anaesthesiology for more than 10 years. So I've been involved in the Helsinki Declaration from the start. I'm also a member of the Patient Safety and Quality Committee in European Society of Anesthesiology and Intensive Care, and in the Norwegian Society. And I'm a paid board member of the Norwegian Standardization Organization. Except for that, I have no financial or commercial interests in today's topics. So the topics of my talk would be the background and presentation of the Helsinki Declaration on Patient Safety in Anesthesiology, some words about building a safety culture, and then on speaking up and communication. So why do things go wrong in medicine? Well, all of you possibly heard about Murphy's Law. And Murphy was working on Edwards Air Force Base in California after the war. He said that if there are two or more ways to do something, and one of those ways can result in a catastrophe, someone will do it sooner or later. And perhaps that's the worst possible time. In the UK National Health Service, they even made some lists about so-called never events. Those are adverse events for which they believe that there are so much routines, protocols, and so on, that if you follow them, it should be impossible to end up with this. But still, things happen. And let me start with discussing the difference between something being complicated and something being complex. As you can see, this is the moon. And if I ask, is it possible to travel safely to the moon and back? You would say, well, we did that 50 years ago. But it isn't easy. It's very complicated. But given enough resources It's possible to calculate and to make perfect analysis of how to go and so on. On the other hand, the weather, it's impossible to 100% certainty to forecast the weather here in Bergen in 3 days' time. And it's not because we don't have enough data or enough computer power. It's because the weather in itself is a complex system, which means that it cannot be forecasted 100%. And the same applies to the world stock markets, to some systems in the immune system, in our bodies and so on. And healthcare should be viewed as complex. And we know about how complex systems fail, because on one hand, they are intrinsically hazardous systems, but they are heavily and successfully defended against failure. But catastrophe is lurking just around the corner. And one of the challenges is that this risk will develop all the time. So there are latent risks that will surface now and then. And post-accident attribution accident to one root cause is fundamentally wrong, because hindsight biases post-accident assessments of human performance, which is very difficult. And as humans in the system, we have dual roles, both as producers of those failures, but also as defenders. So as practitioners, we are constantly both making this system more safe by creating safety, but we're also breaching safety rules and so on. And in a way, we gamble on this to a certain degree. So success and failure must be seen and understood as the two sides of the same coin. It's resulting from the same system. It's not that the bad results, they are coming from one system and all the good results from another. It's from the same system. At the same time, we are giving a patient in my hospital a fatal dose of one drug. Across the corridor, we could be saving a baby of 290 grams. So success and failure, the same system is producing them. And all the time, we will also balance the chances of success against the risk of failure. That's our job. And the choices depend on experience, and experienced experts, they would not jump to a conclusion, but they would make a decision fairly soon. And then they would start looking for something that could make them reconsider perhaps another solution, instead of sticking to the solution and trying to find anything that confirm it and kind of neglect other information. Hollnagel made this illustration of this efficiency thoroughness trade-off, the ETTO principle. And according to him, if we are too thorough, this scale would tip to the right side, because the action would come too late, so it would be a failure. And on the other hand, if we are too efficient, running too fast, making too many patients through the system, we will sooner or later do the wrong action and fail, because we are too efficient. So balancing efficiency and thoroughness is the key. And most of the time, we are pretty good at that in healthcare. Some words about anesthesia in Europe, because as a specialist in anesthesia and intensive care, I've been raised in this specialty, and it's developed as a separate specialty in Britain back in 1932. And the UMS was established more than 50 years ago. We have the European Board of Anaesthesiology. On a European level, we are discussing how training should be and and the duration of training and so on, standards for that. And we have the European Society of Anaesthesiology and Intensive Care, SAIC. They are organizing annual conferences, Euro Anaesthesia, and we have the Confederation of European National Societies of Anaesthesia, the CENSA, which is a subgroup of the World Federation, and we have the European Academy of Anaesthesia. So a lot of bodies are involved in this. And why has anesthesia done this? Well, it's because anesthesia was dangerous. And one started to see the value of marking oxygen, because lack of oxygen was one problem early on. And many notable individuals made a lot of cultural developments within our specialty. The Association of Anaesthetists of Great Britain and Ireland, the AAGBI, Safety Committee was established back in 1976, and we had some proposed minimal monitoring standards published back in 1987. And back in 1950, the mortality due to anesthesia was calculated to be about 1 in 2,500, while today, when we are operating on sicker patients and older patients and perhaps more advanced surgery, it's down to 1 in 100,000. And this is a dramatic improvement in patient safety within this specialty, but it has involved standardization, technology and training, and also kind of acceptance of human factors as an important part of safety. And as I said, I've been the Norwegian member of the EBA for more than 10 years. And back in 2009, we saw the need for trying to come up with some European or international agreement on what should be the kind of minimal standard, what should be expected from our specialty when it comes to patient safety. And on the background for the Helsinki Declaration on Patient Safety and Anaesthesiology, it writes that we have a shared responsibility for quality and safety, anesthesia at large. And in my country and in many other countries, anesthesia is not only putting people or patients to sleep, it also involves intensive care, emergency medicine, and pain medicine. And also the whole period around surgical procedure or perioperative process. And during the pandemic, we've seen all over the world that anesthesiologists and anesthesia as such has been one of the key specialties involved at the forefront, taking care of all the enormous numbers of patients with respiratory distress. So the declaration was launched in Finland during the Euro Anesthesia meeting in June 2010. And very early on, lots of national societies were eager to sign on because they found that this Helsinki Declaration was something that was easy to understand. It consists of 8 heads of agreement and 7 principal requirements. And it provides a practical advice on what to do to try to improve patient safety and how to do it. And every anesthetist in Europe are able to do and to improve some aspects of our practice by applying the principles from the Helsinki Declaration. And the 8 heads of agreements, as you can see, involves the patients and the relatives. It's about education. It's about human factors. And also, as it says, industry plays a role because in the design and interface of man-machine, it's possible to build in a lot of safety. I apologize for this busy slide, but it's just to show what you can find in the Helsinki Declaration regarding the actual things to do. I mean, to have protocols on a number of conditions, to use the safe surgery checklist, to try to report annually on patient morbidity and mortality, and to start some sort of incident reporting and looking into what happens. And we knew that from the launch in 2010, the word quickly spread, and a number of colleagues and societies all over the world were very enthusiastic about it. And numerous societies wished to adopt and set up their own signing ceremonies and inviting politicians and so on. And I must pay tribute to colleague Jannicke Mellin Olsen, who has been traveling all over the world trying to, at these meetings, trying to sell in the and spread the message of the declaration. And as you can see here, except for Antarctica, the Helsinki Declaration has been signed on every continent. The European Board of Anaesthesiology back in 2015 started trying to see if we in the EBA could do something to kind of make the Helsinki Declaration and all suggestions be reality. And we came up with a publication on recommendation on minimal monitoring, sirens labeling, because medication safety is important, and sedation practices. We set down a patient safety committee, and we came up with some spot on practical advice on medication safety. And here's a checklist, which I think is quite useful for any department to look through and see if there are any areas where they perhaps should see if things could be improved. So what happened after signing up on a declaration is easy work. What about to make some changes happen? And 2 years ago, the Lancaster Patient Safety Research Unit and the Patient Safety Committee in the SAIC proposed a project to see how the Helsinki Declaration was followed up. And the results were published 2 years ago. And what we found was that when it comes to monitoring standards, the levels were very good. And also, most colleagues would use the safe surgery checklist, and only 1 in 20 didn't use it. On the other hand, only 1 in 3 said that they made annual reports on the level of implementation of safety measures, which is something that could be improved. And then 2 years ago, also the WHO arranged the first World Patient Safety Day. And that was patient safety, a global health priority, because safety is as important in the less affluent areas of the world as in our part of the world. And the slogan was "Speak up for patient safety." Also, at the evening of September the 17th, lots of buildings were floodlit in orange to mark the event, which is today. Last year, it was health worker safety, a priority for patient safety, reflecting the challenges regarding the pandemic. And the slogan was that safe health workers result in safe patients. And again, speaking up is important. And Valentina has already shown you some from this, the WHO publication on how to eliminate avoidable harm. And in that book, it says that a safety culture which is essential for achieving this goal. It must be embedded, safety must be embedded into the attitudes, beliefs, values, skills, and practices of healthcare workers, managers, and leaders. It's a broad aim, and it should also intertwine with the overall organizational philosophy and culture. And the elements of a safety culture has to do with clear leadership and commitment, transparency, and open and respectful communication, the ability to learn from errors and best practices in other institutions and organizations, and also to make the balance and the fine balance between a no blame policy on one hand and accountability on the other hand. But as you all know, culture eats strategy for lunch. So it's difficult. The EBA and the Safety Committee in the SAIC published this paper in the European Journal of Anaesthesiology last year, and it was a 10-year anniversary of this Helsinki Declaration. And I contributed one part of that about speaking up as a vital part of safety culture. And the thing is that when we're analyzing adverse events, it's quite often that when you discuss with people who were there, that one or some of them said that, well, I knew that this was going to happen, or I had some information, but I thought that everyone else also knew it. So they didn't speak up or didn't tell anyone about it, or they were afraid of doing it. And there are many reasons for not speaking up. But of course, being able to speak is part of communication. And as human beings, we are all experts on communication, because we feel when someone is speaking to us, if the person is respecting us, if he or she is doing it in a nice way, or if they are threatening, making fun of us, or things like that. And some of you probably know this Dr. House, and he's supposed to be an excellent doctor, but it's all about him. And he makes a lot of fun of his colleagues and try to make them look or sound stupid, because he's the only brain that makes the correct diagnosis and so on. So I think being part of his team isn't a good experience. And the reason for not wanting to speak up when we are in a team can be many. And of course, there are some differences in hierarchy. It's not as easy for a nurse or student to address a doctor. Could be respect for the more experienced colleagues or professions. As a junior doctor, I'm not supposed to tell a senior consultant that he or she just were doing something that I feel wasn't, or that I've learned at school wasn't safe practice. It could be the fear of damaging good relationships. I mean, I'm the colleague of someone, we're even friends, and when he or she is doing something that they shouldn't have done, I'm reluctant to tell them because I'm afraid of losing their friendship. It could be fear of retribution or punishment. If I speak up, that might result in problems for me later. Some of us just don't like conflicts, and we like to be nice people all the time, and we avoid raising concerns and so on. Someone else could do that because I'm just a nice guy. And it could be negative evaluation that we are afraid of that, because I really want to continue working in this unit. And if I start making fuss of this or that, that could result in a bad report, and I'm transferred to some other place. And it also could be that, well, I'm not an expert. I just think that perhaps, so I'm not sure. And perhaps I should wait and not raise the question because I might be wrong. Or it could be that, well, I tried to do it before, it doesn't work, it doesn't help, why bother? And also I think that in today's society, especially in my part of the world, conformity is one reason, because we are afraid of sticking out from the rest of the group. And if nobody else has spoken up about this, then perhaps it's just me feeling a bit odd about this. And it could also be that I don't know how to raise the issue. I don't know how to say it, or I don't have time for doing it because of time pressure. And it's about culture, that in our department, we don't discuss dirty underwear. It's not common to make such comments. It's no use doing it like that. And there seems to be a relation between larger and smaller organizations, in which the smaller ones are more likely to get people to speaking up than the larger ones. And how to encourage people for speaking up. And as seniors, we can encourage the younger team members to do it by being a good example. As I said, smaller organizations and units, it's perhaps from the onset a bit friendlier. It's not that huge, it's not that big. So it could be easier to have, and also to understand the communication lines and so on. And of course, the organization might invite, say, if you hear, we would very much like you to tell us. And we would celebrate rather than punish you for it. And it's about colleagues, being able to make networks between colleagues, juniors, seniors, so none of us may end up as solitary colleagues sitting, eating lunch for ourselves like that. And also to have an active approach against any unwanted hierarchies. Some organizations have tried the possibility of anonymous reporting, and also to openly say that in our organization, we won't retaliate and we would guarantee you that this shouldn't be used against you. It's difficult, but it's about human beings. So in conclusion, we must bear in mind that although we all want to make healthcare as safe as possible, it's complex and it's difficult, because modern medicine is complex and inherently dangerous. We have to get the frontline personnel involved, because those who are doing the actual patient treatment or meeting the patients, they are the experts on what they're doing. If it's possible to standardize and harmonize, that's good, because then it's easier for personnel to get adapted to the environment if you change location or something like that. And systematic education and training for staff, both the junior and the senior and the medical students, is important. And using simulation training is effective here. And it's also important to try to both encourage and also perhaps train personnel in the skills, communication skills, how to raise a concern, how to do that in a way that shouldn't be experienced as a threat or as a bad comment, when it's really about improving safety. And also using patients and relatives, as we've seen, is important that way. But don't expect miracles. It's hard work and endurance, which is needed. I'd like to acknowledge both David and Janneke for valuable input here. And thank you.
Thank you, Dr. Bratebo, for your presentation. Thank you. And now I will leave the stage to Dr. Donat Spahn for implementing patient bed management program implications on patient safety. Please.
Thank you very much, Valentina. I share my screen. Dear colleagues, it's a great pleasure and a great honor for me to speak at the Day of Patient Safety to all of you. And here it is. So I talk on patient safety in acute care setting, the role of point of care coagulation testing, implication for clinical practice. As Valentina already said, I'm the chair of the Department of Anaesthesiology at the University Hospital of Zurich, Switzerland. And we are following and have implemented all of what I'm going to present. First of all, my conflicts of interest, which are no conflicts. It's just a summary of my activities. I do consult companies. I am the co-chair of the European Trauma Treatment Initiative. I do consult universities, the US Department of Defense, and professional societies. Also, I have lots of interactions with companies and with foundations. So let's start with coagulation. And if we say coagulopathy or bleeding, even worse so, but if you tell a physician in a remote city somewhere on this planet about the bleeding, then many think, well, then we need to do something about the bleeding or against bleeding, and we need to do something about coagulation. And many then think, well, then we need to give plasma. And this is so wrong that not even the contrary could be true. That's simply wrong. And there is even a Cochrane database analysis on the efficacy of fresh frozen plasma for cardiovascular surgery. And you all know that the Cochrane analysis usually come up with the conclusion that nothing is so clear and further studies are needed. Here, in contrast, they are very clear, and they say that FFP was inferior to preventing RBC transfusion with an odds ratio of 2.5 versus control. So this is clear. By giving FFP in cardiovascular surgery, you make things worse. It's not only it's inefficacious, you make things worse. And, you know, first of all, these are the side effects or the dire consequences of giving FFP to patients. So mortality goes up, multi-organ failure is more frequent in these patients, infection rate goes up, lung problems go up, and again, it's highly and totally inefficient to treat coagulopathy. There are dozens and hundreds of publications that prove these points. So I will show you here a very short version of why FFP is dangerous and is contraindicated in most clinical situations. Because in many clinical situations, the problem of the coagulopathy is present, because bleeding is not necessarily the coagulopathy. Bleeding can also be simply due to the fact that there is a hole in a vessel, so it's a surgical issue. But apart, if it is a coagulopathic situation, in many situations, trauma is a very good example. You know, it's a problem of fibrinogen. That's the problem. It's not some undefined problem. It's a fibrinogen problem. And if we want to solve the fibrinogen problem, we have to give fibrinogen. And so there are different sources of fibrinogen. Yes, of course, there is fibrinogen in the FFP, but there is fibrinogen concentrate or cryo with very high concentrations of fibrinogen. The fibrinogen concentration, according to this study by Jerry Levy, is about 2 grams, and it's highly variable. And in many countries, there is pathogen inactivation introduced in the last years, which decreases fibrinogen concentration from around 2 grams per liter down to less than 2 grams per liter. We have looked at this in Switzerland. And the clinical consequences of giving FFP in the bleeding patient is that fibrinogen concentration is about stable or may even go down. What's certain is that due to the dilutional effect of the FFP administration, the hemoglobin concentration goes down. And therefore, the uneducated physician then reaches out for an RBC transfusion. But the RBC transfusion, doesn't contain any coagulation potential. So the coagulation potential due to the RBC transfusion goes down further. And Dr. Stupid doesn't know to do anything else but give further FFP. And this results in a vicious cycle. And I can guarantee you that this is happening every day in every hospital where no clear understanding of the coagulopathy in different surgical or perisurgical areas is present, where no algorithms in terms of what to measure and what to do if you find X, Y, Z exist. And this is exactly what we have heard so far in terms of patient safety. You need to have clear standards, clear measurements, and clear indications what to do. And this is what we did with the European guidelines on the management of major bleeding of coagulopathy following trauma. And in 2019, we published the 5th edition already. And as of yesterday, these guidelines have been cited scientifically 2,400 times and have been downloaded from the original homepage of critical care more than 738,000 times. So worldwide, since this is only representing about 30% of total downloads from multiple other sites, these guidelines have been downloaded more than 2 million times, and they have really made an impact in trauma treatment. Again, they provide a framework of what to do, and they set standards. So first of all, regarding coagulation monitoring, what to measure is clearly that we recommend that routine practice include the early and repeated monitoring and hemostasis using a combined traditional laboratory or point of care viscoelastic methods. Now, very important, it's early and repeated. As soon as the patient enters the trauma bay, coagulation must be evaluated, and this can be done either in the lab, then it's very important to measure fibrinogen, or by point of care testing with viscoelastic methods, rotational thrombelastometry or thrombelastometry, either or can be used quite successfully, or you can combine. We come back to that. But it's important to do that immediately to find out if there is coagulopathy, and if yes, what is the cause of coagulopathy. Now, in terms of antifibrinolytic agents, we are very clear. This is one of the relatively few grade 1A recommendation. So we recommend that tranexamic acid be administered to the trauma patient who is bleeding or at risk of significant hemorrhage as soon as possible and within 3 hours after injury. We suggest to give first 1 gram as a bolus, followed by a 2nd gram as an infusion over 8 hours. And since it is so important to give the tranexamic acid early, we recommend that protocols for the management of bleeding patients consider administration of the first dose of tranexamic acid en route to the hospital or at the site of injury. In Switzerland, we moved to this 5 years ago, and I can tell you this made a huge difference. We are not seeing any patients anymore after severe trauma in the trauma with active fibrinolysis, for example, the patients, they come in now with the same type of injury in terms of coagulation, much, much better than before we started to give all patients with significant trauma a first gram of tranexamic acid at the site of injury. So this is something I really would like to urge you to implement as a standard in your countries. So, and what do we do then initially? Initially, we offer in the European trauma treatment guidelines 2 strategies. You can give initially, but only initially, FFP and RBC in a 1:2 ratio, or much better, you give fibrinogen if needed and RBCs as needed. But this is only in the very first minutes. Afterwards, we highly recommend a goal-directed strategy. So this is, you measure in the lab or with viscoelastic testing what is going on in terms of coagulation, and then you know what's the main problem, and then you act to solve the main problem. And then after this, you re-measure the coagulopathy again to make sure that you improve, or the patient improved in terms of coagulopathy, or the coagulopathy is gone, hopefully. And these agents you give, some of them are expensive, some of them are having also unwanted effects. So you want to give the right dose, but not more than the right dose, and you want to be sure that the success you wanted to achieve is achieved. And as you all know, that the situation after trauma is very dynamic. So after a certain while, let's say 20, 30 minutes, that you do these tests again to make sure that everything is still okay. Or maybe you need to re-intervene or do something else, but always goal-directed. Now, in terms of fibrinogen, we we recommend, because we are sure that this is right, a fibrinogen level of at least 1.5 grams per liter, or to treat viscoelastic signs of functional fibrinogen deficit. So we did this study here and compared a first period where everybody did a traditional management without guidelines and algorithms, with a second period with a goal-directed coagulation and transfusion protocol according to European Trauma Treatment Initiative. And the outcome of this study was the observed versus the TASH predictive massive transfusion rate until ICU admission, the use of blood products, and clinical outcomes, including mortality. And this was the coagulation algorithm that we used. So if the FITTEM had an MC, a maximal clot firmness of less than 7 millimeters of mercury, 7 millimeters, we gave fibrinogen 2 to 4 grams IV. If we have detect signs of fibrinolysis, we add tranexamic acid. If we have a factor of 13 below 60%, we give factor 13. If we have a platelet count below 50,000, then we give platelets, or if the XTEM or INTEM, the MCF is below 40 millimeters, we also give platelet concentrate. If the factor 5 is below 20%, then we give FFP. This is the only indication for FFP. Okay, and you can see here that the TASH predicted massive transfusion rate in the first period was 12%, and we observed exactly 12%. So this means at this time, we were as bad as anybody else on this planet. In the second period, due to the fact that the type of trauma changed a little bit, the expected massive transfusion rate was 7.5%. The observed observed was just about half, namely 3.7%. So this time we had a higher success in avoiding massive transfusions than the rest of the planet. In terms of blood products given, you can see here RBCs until 24 hours after hospital admission. Instead of 5 units, we were down to 2. FFP, instead of 3, we were down to 1. Platelets were about the same. And the FFP to RBC ratio was also reduced. Now, in terms of clinical outcomes, hospital length of stay was about the same. ICU length of stay was -3 days. This is highly relevant. And ventilatory support was down from 7 days to 3 days. And this is tremendous. And this is, I'm sure, I cannot prove it, but I'm sure this is due to the fact that we gave hardly any FFP anymore. And see here, in-hospital mortality was down from 33% to 22%. So following such guidelines reduces important clinical outcomes, including in-hospital mortality. Now, the National Institute for Health Research, the NHS in Great Britain, is very famous for their health economic evaluation of different technologies. So they looked at the viscoelastic tests, and they came to the conclusion that, of course, their use in accordance with algorithms reduces the use of RBCs, FFP, and platelets. But in terms of costs, there is cost saving in cardiac surgery, not a lot, but a little bit. And see here, cost savings in trauma, £700. So these viscoelastic tests, ROTEM or TEG, they're not expensive tools, but they help saving money. They help saving the use of allogeneic blood products, and they improve clinical outcomes. This has been shown many, many times. Now, let's go to cardiac surgery. Of course, you know, if there is bleeding, first of all, usually it's bleeding due to holes somewhere, and bleeding requires a mechanical action by the surgeon and has a priori nothing to do with coagulopathy. Only about 20–25% in some situations, bleeding in cardiac surgery is due to coagulopathy. Klaus Gerlinger, who is present today in the webinar, they did a first groundbreaking study in 2011, published in Anaesthesiology, and they compared the period before versus the period after the introduction of a ROTEM-based coagulation algorithm in their institution, and they looked at 3,800 patients. The outcome was transfusion of blood products and the incidence of massive transfusions. And this is one part of their algorithm. So after the declamping of the aorta, they do a ROTEM, and if the MCF is 0, then of course in the FIBTEM, then of course this is a problem of fibrinogen. So then they gave 50 milligrams per kilo fibrinogen concentrate. If the A10 in the XTEM and is below 30 and the A10 in the FIBTEM is above 6, so it's a platelet but not a fibrinogen problem. So then they order platelet concentrates. Besides, and very important, they always looked at the optimized the milieu interior. So they avoided hypothermia, low calcium, acidosis, and the very low hemoglobin. And when doing, and then afterwards, you know, this is the algorithm. We don't go here into details, but they simply depict what can be measured and what does this mean. And if it's, you know, a residual heparinization, of course, repeat dose of protamine, etc., etc. And very important and marked here in red is re-examine Rotem, multiplate, or whatever measures you do in your algorithm after the treatment to make sure that you have achieved the desired effect. And here you can see that, you know, the blue is the active period where they did Rotem and they had the algorithm. So any allogeneic blood products was reduced, RBCs were reduced, FFP tremendously. You see again, nearly no FFP given anymore, which results in the conclusion that FFP is really not necessary. More FFP has been given, more PCC. Now, at least as important was that massive transfusion was reduced into half, re-exploration was reduced into half, and the composite thrombotic adverse events were reduced into half. So very important clinical outcomes. A few years later, or a year later, they did a prospective randomized trial very similar to the first one. You see here, Klaus Gerlinger was again part of the study team. This time this was done in Frankfurt and Essen, prospective randomized trial in patients undergoing cardiac surgery with diffuse bleeding after heparin reversal. And then one group was conventional coagulation management with 2 algorithms, intra and postoperatively, and the other one was an algorithm based on Rotem and coagulation factors. And they wanted to include 2 times 100 patients, but then in an interim analysis, the study was terminated early after 2 times 50 patients, total 100 at the planned interim analysis. And you can see here why the IRB stopped the study, because the Rotem treated patients, they had, you know, they instead of receiving 5 units of RBC, they only received 3. In terms of FFP, instead of 5, they had 0, which is certainly correct. Now, platelets were about the same, tended to be lower, or now was significant, but this is the median, but the distribution was such that this was statistically significant. Fibrinogen usage was the same, PCC also, and these patients that were goal-directed treated, they had much fewer of them received recombinant factor 7A. Now, this is great. But then again, some say, well, but this is so expensive. Rotem is expensive. No, this is wrong. Rotem and the factor concentrate are not expensive. They cost about half as the chemotherapy costed in the conventional arm. So it's super cheap. It costs half. And you give really much fewer allogeneic blood products. And bleeding post-operatively is less. And even more important, survival over 6 months was significantly higher in those that were treated with Rotem and the coagulation algorithm. So this is now a meta-analysis on the benefits of using thrombelastometry-based, Rotem-based coagulation management cardiac surgery with 8,000 patients. And you can see that that mortality tended to be lower, but acute kidney injury was -23%, thrombotic event about half, re-exploration down to about half, transfusion -37%, you know, with all blood products being significantly reduced. So this is a great achievement of this type of coagulation management in cardiac surgery. So my conclusions are that coagulopathy is frequent after major trauma and also in cardiac surgery. Immediate viscoelastic coagulation monitoring is key for a successful individualized goal-directed coagulation algorithm. And the compliance with European trauma treatment guidelines or viscoelastic measurement-based coagulation algorithm in cardiac surgery, major surgery, including cardiac surgery, improves survival. What you need is viscoelastic coagulation monitoring and an individualized goal-directed coagulation algorithm. Then you can achieve these success. Thank you very much for your attendance.
And thanks to you, Dr. Spahn, for your presentation. And now I will leave the stage to Dr. Dinesh Yadav, and his topic will be the concerns in regards to patient safety in the day-to-day clinical practice in acute care setting. So, Dr. Yadav, the stage is yours.
Okay, thank you. It's not being presentation. Okay, thank you very much, Valentina. Thank you very much. Very good morning, good afternoon, good evening to everyone, wherever you are. I'm Dr. Dinesh Yadav this side, and I welcome all the participants to this webinar. And the title of this webinar is The Concerns in Regards to Patient Safety in Day-to-Day Clinical Practice in Acute Care Setting. So the key points are patient safety concerns and concerns of acute care setting emergency departments. This is the institute where I work. It's in the capital city of India, New Delhi. So my flow of discussion will be related to patient safety. Why is it so important to be discussed? The burden of concern associated with it, the major challenges and concerns of emergency department in acute care setting, diagnosis and problem associated with it, and the role of point of care testing in healthcare. And why patient safety is so important? Because it is recognized as a serious global public health concern. And the idea is no one should be harmed while receiving healthcare. To mark the significance of security of the patient, first World Patient Safety Day was celebrated 3 years back, that on this date only, that is 17th September 2019. It was started by WHO to bring the awareness about patient safety as a global health priority and to ensure the safer healthcare. But the burdens are 10% of the patients are harmed while receiving hospital care. More than 50% of these harms are avoidable. The occurrence of adverse events are very huge, which results in 2.6 million of deaths annually due to unsafe care in the hospital. 25% of patients are harmed in primary care, OPD care, the ambulatory care, and majority of it are preventable. Overall, these factors actually increases the length of stay of the patients in the hospital, and it increases the number of admissions, which leads to the financial burden as well, like medication error. Unsafe medication practices are the leading cause of avoidable harm, which affects millions of patients and cost billions of US dollars. 15% of the total hospital expenditure is due to the adverse events and corrective measures for the rectification of harm due to adverse events. The total cost of harm in OECD countries is estimated to be trillions of US dollars, which is very huge amount. And we know the majority of the region, majority of the adverse events are due to avoidable reason, preventable reason. And we know that the cost of prevention is much lesser than the cost of treatment due to harm. And there could be many reasons, but human factors are the major contributor to the adverse events in healthcare. Human error is nothing but it's part of our human nature. Error is inevitable, even for the doctors. There could be many reasons like inadequate resources, selection of poor procedures, poor working conditions, sometimes staff shortage, lack of appropriate supervision. At personal level, fatigue, illness, burnout, stress, these things actually decreases the ability and performance of the individual. This is very interesting slide, which shows the comparison between the aviation and nuclear power industries with preventable healthcare. The importance of human being are recognized for a long time, and aviation and nuclear power industries are perceived as high-risk industries, but it has only 1 in 3 million risk. Compared to the preventable healthcare, here the risk is very high, 1 in 300. So the aviation and nuclear industries have much more better safety than the healthcare. You must be wondering why there is so much of difference. I personally believe that healthcare system is an increasingly complex system. It has to consider so many variables, like whenever we are considering any patient, age, sex, weight, different pharmacokinetics, pharmacodynamics, the comorbidities. So healthcare is actually probably the most complex of all the human endeavors. We cope quite well with the complexity because the human brain is very powerful and very flexible. But being human, we can misperceive the situation despite the best of our intentions. Being human, we are susceptible to make some silly mistakes. This is one example of the traps in healthcare, like look-alike and sound-alike medicine. In this picture, 2 medicines are there. The appearance of both the medicines are very similar to each other, but the composition is different. The name is different. The indication, the use is different. But sometimes it is dispensed incorrectly to the patient, which harms. Now we'll come to the concerns of acute care setting, the emergency departments. The scenario of emergency department is like the work processes, the workload and the work pace is often very high. And it can change rapidly because the nature of emergency department is unpredictable. For the number of attendance of the patient or different presentation of the patient, sometimes different priority of the patient condition. Secondly, crowding. Crowding is considered as a threat to patient safety. It increases the length of stay of the patient in the hospital, morbidity and mortality. Another factor is multitasking. Multitasking occurs very frequently in emergency department, though I personally believe that clinicians of the emergency department need to master the skill of multitasking to some extent, because it occurs very frequently in emergency department, but it implies risk to the patient safety. Few of the studies have shown that multitasking is directly proportional to the increased risk of errors. Interruption, lastly, the interruption break in the performance of human activities. Sometimes such chaotic situation in the emergency departments are there, like it causes disturbances, distraction, disruptions, and change of task, etc. For clinician, the major challenge is diagnosis of the disease. Whenever the patient comes to the emergency department in any acute care setting, the diagnosis of disease is the priority. It is based on the history from the patient, the clinical examination, the laboratory results, and sometimes the radiological evidences. Once the diagnosis of the disease is made, the prognosis is ascertained, then what are the treatment options are available, that is decided, and outcome probabilities are evaluated based on the patient condition and need. But diagnosis is the most important aspect, and the problem associated with these are any inaccuracy in diagnosis or delayed diagnosis is the most common cause of potential harm, and it affects millions of patients. Diagnostic error is nothing but failure to identify the nature of an illness in an accurate manner and timely manner, which accounts for 5% of the problem, which causes misleading and wrong diagnosis. And half of these errors are associated with severe consequences. So modern day medicine is evidence-based medicine. It heavily relies on the analytical results from the laboratory. So it should not only just be reliable, but it should be very faster. And here comes the role of point of care testing. Point of care testing, in addition to the central laboratory, the main laboratory. Point of care testing, POCT, is when the test is performed near the patient or at the site of patient, with the result leading to the possible change in the care of the patient. The overall whole purpose of POCT is to shorten the time to clinical decision making and overall clinical pathway. It actually, POCT allows the clinician and the staff to achieve the real-time diagnostic lab results using portable analyzer, handy analyzer, easy to operate, and the results are available within minutes than hours. It enables the clinician in rapid triaging of the patient in emergency departments. It helps in streamline the diagnosis, like to rule in some certain condition or rule out a few of the conditions. So it can be considered as very useful tool for most efficient and effective quality patient care. But POCT is not free of limitation. There are some challenges. These are not error-proof. There is lack of quality assurance. Training is one aspect which is a very difficult thing. The quality of POCT depends on the person performing the test, the person interpreting the result. They might not be well trained to do that. Like compared to the central laboratory, the people who work in the laboratory are laboratory educated, trained and experienced to handle the situations. Another aspect is it has limited range of tests available till now. But what is the prospect of POCT? The future is expected to expand as like diabetes has drastically altered the management. Being glucometer is a very useful equipment for diabetic patient. And in recent time, the medical care is evolving very rapidly, and it is becoming more and more consumer focused and patient driven. These days, focus is more towards the early detection and treatment. And in coming times, I hope the innovation will bring more tests to this POCT test menu with better sensitivity and specificity. But I believe that POCT is never likely to replace this central lab, but it will be very useful in emergency department, ICU, sometime in the ward, at primary care, in remote location where access to the central laboratory or main laboratory is difficult. So in this situation, it will be very useful. I have a case study to discuss here. A 4-year-old girl came to emergency department in semi-conscious condition. There was no history of pain, fever, vomiting, loose stool. Only one history was there of skin pigmentation 3 weeks back on right side of her neck. The sample was sent to the lab. which was very lipemic. Look at this picture. This is the 5 tubes are there of first 3 days of the sample received in the lab for this patient. I am using a very good quality machine, high-end machine. But look at the serum here. It is very milky, frothy, very turbid. None of the equipment could give me any result, and there was errors only. So I use the blood gas analyzer and process the sample in the machine. And the results are pH was 6.85, indicated acidosis. PCO2 was very low. Bicarbonate level was also very low. And casual blood glucose level was 404 milligram per deciliter. But I could not believe this result because all high-end equipment failed to give me any result. So I used the conventional method. I brought a pH paper to measure the pH of this sample, which was acidic, of course. So acidosis was in mind. I requested the clinician if he can provide the urine sample, and it actually delayed the process 2-3 hours. But in urine sample, the glucose and ketone bodies was present. So it made the diagnosis very easy. The patient was diagnosed with type 1 diabetes mellitus, recent onset, new onset type 1 diabetes mellitus, insulin dependent diabetes mellitus. The clinician was very happy because there was no such complaint to handle in the emergency department. So intensivist started with simple titration with insulin to this patient, and most of the parameters, almost all parameters came down to normal within a week, except for the triglyceride. The estimated triglyceride of the day 1 sample was close to 16,800 milligram per deciliter. But because of some technical reason, the reported value was 13,800, which is probably the highest reported triglyceride level in a 4-year-old kid. So this POCT was useful for me in making diagnosis of this difficult patient. There are few publications associated with this. Now, how can we enhance the patient safety? When something goes not well or wrong, no blame culture. We should avoid blame culture. We should work on the root cause. What happened? Why did it happen? What to do to prevent such thing? So root cause analysis and work finding out the solution, or identifying the and removing the cause of the problem is the best way to arrive at the solution. Next is the best way to breeze the laboratory staff with the user of laboratory services like clinician and nursing staff is by providing not only the accurate result, which we are doing regularly, but faster way. So which will be useful in making early diagnosis and treatment of the patient. Of course, effective communication is the important aspect because miscommunication leads to many problems. I wish everyone a very happy patient safety day. Thank you very much. Thank you.
And thanks to you, Dr. Yadav, for your presentation. So that I will pass the word to Dr. Malak Al-Mashali from Saudi Arabia for her presentation on the laboratory perspective, patient safety in blood gas testing. Okay, please.
So, hello, can you see the slides?
Yes.
Okay. Good afternoon, everyone. First, I would like to thank the organizer of this meeting for inviting me here today. High reliability in a healthcare means the elimination or prevention patient harm, also the harm to the workforce. That include nurses, caregiver, also the physician. High reliability means zero patients harm. That's why we are all here today to highlight the most critical topic related to the patient safety. And one of the most important points related to blood testing is the pre-analytical error. My talk today is particularly related to the blood gas analyzers as point of care testing and patient safety. I will cover multiple points in this presentation, focused mainly in pre-analytical error and the problem that blood gas sampling user might face during their practice and the options of solving this problem. With regard to the COVID-19 in Saudi Arabia, the health law in the Kingdom of Saudi Arabia aim to provide comprehensive health care for all people in a fair and accessible manners. Saudi Arabia has demonstrated over 30 million doses of COVID-19 vaccine across the kingdom, and over 587 inculation centers have been set up around the kingdom to ensure that all citizens and residents have access to coronavirus vaccine. So let's talk here about the blood gas. Blood gas and pH analysis has more immediacy and potential impact on patient care than any other laboratory determination. The National Committee for Clinical Laboratory Standards informed that in blood gas and pH analysis, an inaccurate result can often be worse for patients than no result at all. This highlights the importance of focusing on the pre-analytical error to ensure patient safety. I'm sharing with you here in this slide some facts related to the medical errors. So the medical errors is a significant contribution causes of morbidity and mortality in the hospital, and it can be identified as the third cause of death after cardiovascular diseases. Diagnostic errors will delay and giving wrong or missed in the diagnosis. And finally, poor quality sample affect test result accuracy. And you can see this in the blood gas sampling. I will mention about this point later in this presentation. So why reducing error is important for patient safety? When it comes to patient safety and reducing errors in point of care testing, it's important to follow a comprehensive systematic approach to ensure patient safety. And this includes to create and review a source of errors, and to identify the potential error in the testing process, which including the whole phases of the testing, pre-analytical, analytical, and post-analytical phase. And also to design method of control resolution for each potential error in every step or phase of the test. And finally, it's important to build and implement mitigation plan. So the pre-analytical error in blood gas testing is the blood gas is commonly ordered in a hospital setting, where the results are often used for critical care and required a rapid turned around time. The pre-analytical error can occur from several sources, starting from choosing the correct test, ensuring that the specimen introduced into the blood gas analyzer correctly. So let's focus here on the source of the major pre-analytical error. As you can see here, the microclot and the air contamination, and also from sample collection phase, from sample handling and transport, and finally from inadequate catheter flush. Let's start here with the first challenge in the pre-analytical errors, which is the microclot. The microclot or fibrin strands are very small and not easily detectable by POCT user. It can be result from improper sample mixing, and it can remain on a sensor for multiple sampling cycles or transfer to another sensor, and can alter the values if not detected and giving inaccurate results. So for good pre-analytical practice, it's important to have immediate and effective mixing to reduce microclot formation. So what is the action and solution for this challenge? First, to have a proper collection device. The ideal collection device for arterial blood sample is the plastic syringe and self-filling disposable. Also, a 1, 3, or 5 ml syringe filled with appropriate type of lympholyzed heparin. And second, having a proper anticoagulant. And finally, good mixing is important to minimize the risk of the clotted blood. The second challenge for the pre-analytical error is the air contamination or bubbles. In practice, the sample exposure to room air for 10 to 30 seconds can cause significant change to the blood gas value. The degree of bias or resulting error is unpredictable, and it can give a positive bias or a negative bias. And there is several factors that affect the blood gas result, as you can see here in the slides. But the most important is how to solve this and how to avoid the air contamination. The operator should remove the air bubbles immediately after collection. And it's crucial to analyze the sample as soon as possible after the collection. Also to remove residual air and recap the sample after analysis. The third challenge is specimen mixing and heparin. Remember that the heparin prevents but doesn't reverse clotting. The microscopic clots can form in a little as 10 seconds. That's why for effective anticoagulation, it's important to ensure appropriate amount of heparin is used and appropriate sample mixing, as sample mixing can affect several results, especially with the hemoglobin and hematocrit sample. So what is the solution for the specimen mixing and heparin concentration? If reporting electrolyte, use low or balanced lithium heparin formulation. Concentration should be above the 20 international unit per milliliter. Don't use liquid sodium heparin for electrolytes. And it's important to use correct syringe size and fill above the 50% for the electrolyte especially. And with regard to mixing, to avoid clots, mix sample immediately after draw while holding puncture side. And for the 3 ml syringe, mix with rocking motion for 30 seconds, as you can see here in the picture. And for 1 ml syringe, require more vigorous mixing. Remix immediately before the sample. So good mixing technique improve the accuracy of the blood gas results. Another challenge is the post-collection metabolism. As you know, that blood cells continue to metabolize in the syringe after the sample is drawn. An excessive delay for analysis and warmer ambient temperature increase the metabolic rate, which resulting in oxygen and glucose consumption, carbon dioxide and lactate production, and decrease in the pH. So how to avoid this challenge? I would like to call your attention here to a very important point, which is the temperature and the effect of the temperature in the post-collection metabolism, as so many pre-analytical errors come from this, especially in the sample collection phase. It's important to analyze the sample as quickly as possible after obtaining the sample to reduce the error potential. Analyze capillary sample within 10 minutes. If the sample analysis is estimated within 30 minutes, room temperature storage is recommended, so no ice needed. For analysis in 30 to 60 minutes after draw, ice ciliary storage is recommended as cooler temperature reduce the metabolism. And after 60 minutes of, after the 60 minutes of the ice ciliary storage, sample might not give you the reliable result. And this leads to increase the PO2 and the potassium. Another challenge operator might face during the sampling and might lead to pre-analytical error is inadequate catheter flush. The indwelling vascular catheter require flushing the solution to keep lines clear, as samples contaminated with line flush solution will have inaccurate values. So how to mitigate this risk? It's important to flush arterial and venous adequately before sampling. Appropriate discard volume is dependent on the dead space volume of the catheter used. The dead space is the volume in the catheter line for the blood vessels to the drawn of the blood. So it's recommended to discard double the dead space volume to ensure quality of the sample. Studies showed that removal double the dead space volume for infusion waste will help assure that reported results are accurate. Another challenge is the capillary sampling. So in the practice, squeezing puncture site can accelerate clot formation. And remember, capillary blood is not arterial. It is a mixture or arterial blood and venous blood, capillary blood, and also tissue fluid. So squeezing the site can compromise the accuracy and promote microclotting formation. Also, the practice of transferring the sample from heparinized capillary tube can alter the sodium and ionized calcium by increasing the heparin concentration. So this type of transfer can also alter the blood gas value from exposure to air. So what is the solution for the capillary sample? Some studies have shown that pre-warming capillary draw site increase blood flow, which facilitate filling of the capillary tube. And also make sure to remove any residual skin cleanser from the site of the sampling. Angle the tube slightly downward while drawing the sample, as this will help filling with no airspace. Then cap both ends of the tube. Be sure to remix before analysis. And finally, occasionally caregivers will draw sample from vascular line for multiple tests and transfer the capillary tube for BG analysis to reduce blood drawn volume. If both syringe and capillary tube are heparinized, the heparin concentration will be increased. This practice has potential to lower sodium and ionized calcium value. So use non-heparinized syringe and with the heparinized capillary tube, in this case, if you report the electrolytes. We are reaching here to another source of pre-analytical error. There are other practices that can improve the result accuracy, such as having a steady state policy, as this allows for equilibration time, which is very important, especially in critical ill patients. Also, pneumatic tube transport of blood gas specimen is not recommended, which lead to increase the potassium from hemolysis and giving inaccurate result for other parameter due to the excessive mechanical force in the pneumatic tube. And finally, if vacutainers are used for ionized calcium analysis, the tube should be completely filled. Otherwise, the partial pressure of carbon dioxide will decrease and causing the pH to increase, and this yielding to a false lowering of the ionized calcium. So now you know most of the pre-analytical error in any clinical practice for blood gas sampling. The key point here are, in order to have a good practice, it is important to have continuous and hand-on training in place. Then assess the level of the user's awareness and practice with the competency assessment. It's important to use educational material in POCT area to notify the user about the importance of the pre-analytical error. And finally, remember, delivering education and training is important in reducing patient harm and ensure patient safety. So in addition to the training and education, compliance with accreditation standards is also important, which will help improve patient safety and minimize the risk by providing a continuous quality improvement effort in the healthcare facilities. And this by following the standards to ensure the quality through the testing process in the blood gas analyzer. So now we have the tools that help us to ensure patient safety, from quality management system to training and education and competency, but without teamwork, always there is a gap. In clinical setting, understanding of the teamwork and task, the equipments and workflow, the communication and culture is very important to make healthcare safer for patient and staff also, and to optimize well-being and improve system performance. So let's focus here on the quality control in the blood gas analysis. The quality umbrella will include the quality management system, the quality assurance, and the quality control. In the blood gas test, the quality solution should mimic the patient sample as much as possible, and all results should be managed as a patient sample. So when the quality control results fall within the expected range, the clinician can be confident about the usefulness of the result generated by this analyzer. Remember, the blood gas is one of the most sensitive specimens sent to the clinical laboratory for analysis. An incorrect result often be worse for the patient than no results at all. Here is how it's looked like in the traditional blood gas analyzers. The quality control run in open system with lots of tubing and pipetting, as you can see here in the picture. Each component bring a variation to the system that need to be controlled. I will go quickly here for the type of quality control in the blood gas analysis. External QC, which will introduce by the operator manually, and the automated quality control, where the quality control solutions or the ampoules are on board of the analyzer, and the system is programmed at specific frequency time, every 8, 12, or 24 hours, depends on the policy of the hospital and lab standards. And the electronic QC, which does not follow the same pattern as sample, only for some parts of the instrumentation. In the three cases, an instrument is commonly controlled at the moment of the quality control solution process. So no automatic intervention action is taken after the quality control. An option available to ensure the quality is to have an IQCP plan, and several resources for information available, and you can check the IQCP document from the CDC for more information. The quality management system is a collection of process covering all the activities of quality process and documents with a corrective action to ensure that's the corrective action on place. So let's talk here about the new technological innovation, which is the intelligent quality control management system. There are some blood gas analyzer in the market coming with intelligent quality control management system, and this option will help to have a continuous quality assurance. So the IQM provides continuous monitoring of all analytical process before and after every sample, provides ultimate assurance. The error detection time will reduce from hours to minutes, and errors are automatically corrected and documented. So you will have a corrective, a corrective action system. And it's help ensuring quality result, eliminating and the maintenance, delivering a cost of efficient outcome and improving patient care. And with this technological innovation, the intelligent quality management in the market moved from operator-performed quality practice to manufacturer-assured quality. So give faster detection of errors. As you know, manually performed quality control practices are too complex for effective use at point-of-care testing, especially when it's performed by the non-laboratory practitioner whose primary focus is the patient care, and they don't have the background about the quality and the lab standards. So having this option will definitely help to correct all the process without missing any errors. As you can see here in the picture, the differences between the continuous quality management versus the traditional quality control. In the IQM, as you can see here, the errors automatically detect, also corrected and documented immediately, which will ensure the quality of the results. On the other hand, in the traditional quality control, either manual or automated, there is a chance for error outside of the window of the quality control, which is not detected. In this slide, I will explain about the transient errors or sample-specific errors, which are errors that occur and cause inaccurate sample result, and these errors can come and go without detection. These errors can affect a single sample or multiple samples sequentially. So let's say you run a sample and the results don't match the clinical picture or don't pass the delta check. What do? You will rerun the sample again. The repeat is perfect, so you will verify these results and reject the first set of the results. You just encountered a transient error. This is the norm. This is the normal in the laboratory and 95% confident interval. So as you can see here, even in the literature, transient error are well documented, and pre-analytics tend to be one of the main contributor of these transient error. Here are some specific literature examples from key opinion leaders that show a trend of ongoing transient errors in critical care testing. These articles highlight the impact of sample-specific errors on patients' results. So how to solve this problem and what is the solution? Regulatory agencies such as CLSI offer guidelines for reducing the error, and you can check the document in the CLSI website. As I mentioned earlier in this presentation, the training and education is very important to ensure patient safety. It is important to design a training session covering all the phases of testing from pre-analytics to post-analytical phase. You have the option of the face-to-face training in addition to the e-learning option. So the POCT users have access to the information needed for their best practice. And also always have a communication channel with the users for them to be competent and well trained. And finally, don't forget to provide them with any updated information for their good practice. Let's highlight a very important point here, which is the cost of the medical error. There have been several publications that have focused on the incidence and cost of avoidable medical error. The pre-analytical errors can occur at the time of patient assessment, or from the test order entry or patient identification, and also from the specimen collection, specimen transport. That's why the focus of this presentation was on identifying the source of pre-analytical error and improving the practice for the blood gas sample, because it's crucial for patient safety, and also it is added burden to our healthcare system with regard to the cost. We reach here to the end of this presentation. And if there is one thing I would like you to remember from today's presentation is when choosing the device, select the device, provide best quality control option with less user intervention. And for the risk assessment, identify the risk according to the selected device and SOP. And for the risk mitigation and solution, develop the risk mitigation plan in your clinical setting. And finally, have a training program and develop continuous training plan to assure the best practice. Thank you for your attendance.
And thank to you, Dr. Al-Mashali, for your presentation. And so we are here now, almost ready for the roundtable discussion. So let me first of all thank you all for your presentation, because even in the chat box there are several questions, comments and compliments to you all for your presentation. And just a brief comment from my side, so that it is clear that the leitmotif of this webinar is not only patient safety, but also using patient safety as the common ground to start the communication and to engage the communication between lab people and point of care people. So between the healthcare professionals and across the hospital. So I would like to, first of all, address a question to our medical directors, so Dr. Klaus Gerlinger and Dr. Mark Rubinstein, because I would like in particular to ask to them a comment, because in the WHO Global Action Plan, there was specifically one action related to the safety of blood and blood products. So if you can comment on this, the need for implementation of operating procedures and programs for transfusion services and hemovigilance programs.
I think it was already mentioned by several speakers that one great step is, first of all, to avoid any harm. And we know that with medical interventions, we can harm. And blood transfusion, which is not necessary, is definitely one point which we can reduce. And I think that's more or less the concept of patient blood management, that we want to keep the blood of the patient within the patient instead of just transfusing blood from outside. And sometimes it starts with very small things. It was mentioned that, of course, we need, when we take a blood sample, 2 times the dead space volume. But nowadays we can use closed systems. So we should not, in particular at the ICU, throw away every hour 20 milliliters of blood from a patient. So there are also ways to assure quality at the one hand, but also to avoid blood loss, which is not needed. So I think this is an important point. And also from Dr. Rattewe, it was mentioned the standardization is something important. So we don't have to invent the wheel every day again. So we can learn from literature. And also Donat Spahn mentioned, it's not, the advantage is not to run a point of care test, to do also the right interpretation. And again, implementing algorithms can be very important. And the last point I want to address is, I think we have to look at implementation barriers. And Donat showed quite nicely that a lot of people think, okay, this is too expensive from my hospital. But when we see that we can reduce transfusion as well as complication rates, it's finally cost saving. And that means, yeah, we have to, I think, to analyze in our own hospital, also locally, what are really the barriers? And it starts with awareness, that people think, oh, that's not my problem. That doesn't happen in my hospital, which is completely wrong, usually. The second is it's too complex. And by implementing algorithms, we can reduce the complexity that even when we talk about coagulation management, even non-hemostasis experts can follow a protocol. And finally, we have to be aware about costs, which are, yeah, driven by complications. And that's, I think it's even a bigger challenge for the hospital. And on the other hand, can be an advantage. So that's my key points from my personal point of view.
Absolutely. And so I would like to ask to Dr. Rubinstein, and in all of in this implementation of, for example, the patient health management program, how can we make the therapy monitoring fit inside this program?
Yes, thank you. So first of all, thank you everyone for great presentations, very useful. From that question, my point of view, there's many technologies out there, right? So it is hard to say one technology will work. I think it's a combination of multiple technologies, multiple things that will work to ultimately get that goal, right? Patient blood management. There are certain things that you can do even before starting surgery, cardiac surgery. A lot of these patients are in a lot of medications that will prone them to be an excessive bleeding. So if there's any monitoring you can do beforehand to assess what the status is of that patient before you go into surgery, also helps determining or reducing the risk of bleeding and transfusion. So that's from one standpoint. From the other standpoint, intraoperatively, there's a lot of other monitorings that you can do. There was a lot of things presented, but there's many things you can also do, other type of testing like ACTs that you usually continuously do to assess the bleeding on these patients and prevent the transfusion. Of course, as we was presented, transfusion is good, but it has a lot of risk, and you're trying to avoid that bleeding. And like Klaus said, there's a lot of things you can also do to implement and to prevent from that waste of blood. I think a lot of our testing do in order to be effective or give you an accurate results, there's a lot of bleeding, a lot of waste that you have to do. And that's something we have to work on to try to reduce that waste and on that end, reduce the bleeding.
Yeah. And I think that overall the role of point of care testing in acute care setting is always somehow reducing those complications that are avoidable and perfectly in line with improved patient care, that is reducing those harms that are avoidable. So just to have the final question that I would like to address to you all, actually, it is something that can be used as how to create this kind of strong connection between the lab and the point of care, because we saw within our webinar that overall, our objective is always to improve patient care and towards patient safety. So I would like to ask, first of all, to Dr. Dinesh Yadav and to Dr. Malak Al-Mashali, their experience and the suggestions on how to create this kind of strong relationship between lab and point of care. Please.
Okay, I'll start. First, you have to think to close the gap between the lab and the point of care users. And put in your mind that POCT users are non-laboratory backgrounds. So they need more education. And as long as you have a well-structured training program, and as I mentioned in the presentation, covering all the phases from pre-analytical, analytical, and post-analytical errors, the users will know more about the test other than only for sure that taking care of the patients is one of their ultimate goal, but you also to have to know the importance of the pre-analytical error to have an accurate results, and this will lead to the patient safety as well. Always close the gap between the lab and by ongoing communication and educational materials.
Yeah. Dr. Yadav, your experience?
I agree with Dr. Masali. She said most of the things. What I do, I shared one case study where I was advantaged by this point of care testing. And the feedback is very important. Sometimes we had blood gas analyzer at different ICUs, nephro ICU, medical ICUs, emergency department. And sometimes the clinician has a doubt that these values are not matching with the clinical condition. So what we prefer to do is, whenever the sample is taken, I exercise the same sample in our main laboratory. And then we compare all the results like electrolyte or whatever. Particularly the electrolyte is the one thing which can be measured with our central lab equipment. And once analysis is done, then we come to the conclusion. And most of the equipment like blood gas analyzers are auto-calibrated kind of thing. So most of the time I found it very satisfactory and good. But what happens, as per the point of care testing machines are concerned, we need to understand that the validation part is very important. And training is an issue because it is point of care machine is not operated by the people who are actually lab educated. So there may be some shortcomings which cannot be evaluated timely by those clinically, they are trained clinically, but not lab educated, experienced with some problems. So whenever it comes to the gap between the point of care testing and lab, and we need to adopt some technology or method to bridge the gap by ensuring that whatever the values produced by the machine are near true. I believe in one thing, that machine or equipment will remain machine. It will give some or the other value whenever we process the sample. Our expertise and skill needs to ensure that the value produced by the machine is near true or not. So it depends on means how do you merge how is the working culture and how we bridge the gap between the user of point care of equipments with the main laboratories.
Yeah, it is again working collaboratively together, lab people and point of care healthcare professional and lab healthcare professional, so that always respecting the peculiarities of their job roles at the end and the differences between job roles and the background. Yeah, for sure. And I would like to ask also to Dr. Span, since also in his experience and in his presentation, he was mentioning, of course, point of care testing, but also some kind of lab testing as well in the protocol for transfusion practice. So which is your experience of the communication and exchange with the lab, between the lab and the point of care?
Yeah, that's an interesting point. We have an intense collaboration since more than 20 years. And, you know, it's staggering how each side, so to speak, sees only their own side initially. So, of course, you know, clinicians like anesthesiologists are not aware of all the pre-analytical details that need to be taken care of so that the results are perfectly valid. On the other hand side, a typical lab person who is really only working in the lab has no clue whatsoever how important the speed of the results is in clinical practice. So we have meetings in the OR with lab people, so they see, you know, how critical the patient is and how quickly we must have the information, the correct information, of course, to treat the patient specifically and according to his or her needs. And the two types of technologies, being lab technologies or point of care techniques, they're not opposing each other, not at all. They're, you know, they're complementing and they can be combined, and they look also at different aspects. So I think that the way we did it is a very long way to create an mutual understanding of the problems of precision, errors, and also the velocity, the speed, until we have the perfect results necessary for an adequate treatment of the patient.
And it is a very good example of exchanging not only knowledge, but also experience. So exchanging the way of working between and taking the lab healthcare professional directly to the OR, it is a very good experience to be shared. So Dr. Bratebo, if you want to share your experience.
I totally agree with my colleagues that communication and mutual understanding of the situation and the problems and the challenges is the key. So I think starting just discussing with each other and the simple suggestions and solutions and advice that you had today regarding arterial blood gas testing, which is a simple but very important test. And also regarding blood management, I think that going through the process and ensuring that all steps are being performed correctly at both sides, and also understanding this complementarity between the lab and the point of care testing is very important. So it's all about communication and understanding, and as has been mentioned earlier, I mean, crowding and multitasking, interruption and so on, are things that the more we are aware of them, the more likely it is that we might handle them as well.
Yeah, totally agree. Yeah. And we are running out of time, unfortunately. Yeah. I would like to ask maybe 1,000 of questions, even more on the topic of patient safety, but I would like to ask for a final comment to Dr. Gerlinger and to Dr. Rubinstein, again, in the matter of communication and exchange, creating the strong relationship between lab and point of care to exchange the knowledge and experience to have a common ground based on patient safety. And I would like to close this roundtable discussion with this final comment from both of you. So please.
Yeah, I can just agree what Donat said. So it was also in Essen, where I was working 26 years, a long way to go. And by implementing or more or less changing transfusion committees to PVM committees, where of course the lab transfusion department is involved, is very important. And also we had the strategy, if possible, that even anesthesiologists are for some weeks in the blood bank or even in the lab and vice versa, so that a lab or transfusion understands if an anesthesiologist says we need it now, which doesn't mean in an hour, that means really more or less in some minutes. And the other way around, that also the anesthesiologist understands that, for example, cross-matching is not possible in 5 minutes. So I think we really have to understand the challenges of our colleagues and of course respect their work. And if we get closer to a common language that we understand each other, this is a good way for increasing patient safety. And my final remark, thanks to Valentina for bringing all this together, and yeah, also improving our communications, as we saw with more than 3,700 participants. I think also that might be a good step forward to increase patient safety around the world. Thank you.
Thank you. So Dr. Rubinstein.
Yeah, so not much more to say. I think everything was covered. I agree with everyone, but I think it boils down to transparency between all teams. A lot of times, some teams don't know what the other people are doing, other teams are doing, or what their process is, and somebody assumed that you can get things done really quickly or fairly easy because in your mind, that's how you're done. So if everyone lays their work and what it takes to get that done, and you set up a pathway or a common way for everyone to work together and everyone understand what the other team member is doing, there's better communication, better transparency, and at the end of the day, better patient management. So that's the only thing I can add at this point, because I think everything else was covered, and I agree with everyone and agree with Dr. Gorlinger. Thank you very much for the invite and for putting this together. It's been great.
Okay. So I will just do the final comments before closing, because I would like, on behalf of Werfen, to thank all of you, all of the speakers, for the amazing presentations. I would like to thank to all the attendees that were here together with us in this patient safety webinar. And so we can say that we have put the first brick to build the patient safety culture today within the Worldwide Patient Safety Day. So again, thanks to all of the speakers, and thanks to all the attendees for attendance. Thank you.
Thank you, Valentina.
Thank you. Bye-bye. Bye-bye.
Bye.
Thank
Bye.
you.
Bye.
Bye-bye.